Purine salvage deficiencies. Purine metabolism involves both de novo synthesis and salvage pathways. Nucleic acids such as DNA and RNA are broken down into nucleotides, then into nucleosides, and finally into free bases. Free bases such as guanine, hypoxanthine, and adenine can either be salvaged or degraded into uric acid for excretion. Ribose 5-phosphate is converted into phosphoribosyl pyrophosphate, PRPP, by the enzyme PRPP synthetase. PRPP then participates in both de novo synthesis of nucleotides and in the salvage pathway. Hypoxanthine guanine phosphoribosyl transferase, HGPRT, converts hypoxanthine into inosine monophosphate, IMP, and guanine into guanosine monophosphate, GMP. Adenine phosphoribosyl transferase, APRT, converts adenine into adenosine monophosphate, AMP. If salvage fails, excess bases are degraded. Hypoxanthine and guanine are converted to xanthine by xanthine oxidase, XO, which is then further converted into uric acid. In most mammals, uric acid is degraded further into allantoin by urate oxidase, but urate oxidase is absent in humans. Instead, uric acid is excreted in urine. Clinically, drugs such as allopurinol and febuxostat inhibit xanthine oxidase to reduce uric acid production, while rasburicase, a recombinant urate oxidase, is used in tumor lysis syndrome to break down uric acid into allantoin. Some drugs target the salvage pathway enzymes. Cladribine and pentostatin inhibit adenosine deaminase, ADA. Specific disorders. 1. Adenosine deaminase, ADA deficiency. Adenosine deaminase is required for degradation of adenosine and deoxyadenosine. If ADA is absent, adenosine builds up, leading to increased deoxyadenosine triphosphate, DATP. High levels of DATP inhibit ribonucleotide reductase, preventing DNA precursor synthesis. This particularly affects lymphocytes, leading to severe lymphopenia. ADA deficiency is one of the major causes of autosomal recessive severe combined immunodeficiency, SCID. 2. Lesch-Nyhan syndrome. This disorder results from a defective or absent hypoxanthine guanine phosphoribosyl transferase, HGPRT. Without HGPRT, hypoxanthine and guanine cannot be salvaged into IMP and GMP. Instead, they are degraded, leading to increased purine synthesis and excessive uric acid production. This is due to compensatory upregulation of PRPP aminotransferase activity. Lesch-Nyhan syndrome follows an X-linked recessive inheritance pattern. Clinical findings include intellectual disability, self-mutilation and aggression, hyperuricemia leading to gout, orange or red sodium urate crystals in the urine, sand and diaper, dystonia and macrocytosis. A useful mnemonic for HGPRT is hyperuricemia, gout, pissed off, aggression, self-mutilation, red-orange crystals in urine, tense muscles, dystonia. Treatment involves allopurinol or febuxostat, which inhibits xanthine oxidase to lower uric acid levels. Genetic code features. The genetic code has several defining features that explain how codons specify amino acids. The code is unambiguous, meaning that each codon specifies only one amino acid. It is also degenerate or redundant, because most amino acids are encoded by multiple codons. The wobble hypothesis explains this redundancy. The first two nucleotides of a codon are essential for recognition by the anticodon, but the third nucleotide can vary or wobble and still code for the same amino acid. However, there are exceptions to redundancy. Methionine, coded by August, and tryptophan, coded by UGG, are each specified by only one codon. The genetic code is commonless and non-overlapping, meaning it is read continuously from a fixed starting point without punctuation, and codons are not shared or reused. The only exceptions to this rule are seen in certain viruses. Finally, the genetic code is described as universal because it is conserved throughout evolution and used by almost all organisms. However, an exception exists in mitochondria, where some codons can specify different amino acids than they do in nuclear DNA.